Good afternoon. Thank you all so much for coming. It's so great to see you here. This is a really special moment for me. I have the opportunity to launch Peapod Bio at the SLAS 2026 conference. This is an organization that means so much to me, and to do it in Boston, Massachusetts, my hometown. Those of you who don't know me, I grew up not too far from here. That's why sometimes I sound like I'm in a Dunkin' Donuts commercial. But honestly, I couldn't have written this any better. And because of how exciting this is, I really wanted to share something special with you because there are so many things that I could tell you about. I could talk about some of the partnerships that we've established, folks like at Enamine, bringing in world-class libraries for our clients to screen. I could talk about some of the assays that we've developed that have launched drug discovery programs on some truly challenging targets. Or I could talk about how we built this company from nothing to being fully operational, delivering data in a matter of weeks. That is a truly remarkable accomplishment and a testament to what this team can do. In fact, we work so well together, one could say we're like peas in a pod. And so to get some inspiration, I was looking at the SLAS 2026 event planner, and I noticed that Hamilton was sponsoring the talk after us. And I thought, Hamilton, how would Lin-Manuel Miranda present a startup or a company like Peapod Bio to this audience? I know, different Hamiltons, but just go with it. And I thought he'd probably start by posing a question. How does a startup in the trenches, lean and relentless, trained at the bench, make a scene, swing for the fences, built to run screens with assays so inventive, and a strong value prop to offer up as an incentive? But I wasn't going to get Lin-Manuel Miranda here. Instead, you have me. My name is Zack Gurard-Levin, and I am the founder and CEO of Peapod Bio. And today, after I introduce the company, I'm going to tell you about some creative solutions that our team has developed using innovative surface chemistry that allows us to detect analytes that until now have been undetectable by mass spec. At Peapod Bio, we know the challenges of discovery outsourcing. You wonder how much rigor your CRO is enforcing. And every day that your data is delayed, that's up to $1 million your company isn't getting paid for the drugs that you're delivering to a sick patient. And it's tough to sit by and wait and be complacent. You need resources, convenience, technological advantage, cutting-edge science without having to micromanage. At Peapod Bio, delivering unrivaled solutions is our mission, and becoming your preferred partner, that's our corporate vision. To ensure data quality, our pre-assay consult is when we listen to your goals, your timelines, and any other condition. We may have ideas to share. With your permission, we'll spend a day designing assays, conditions we envision. There may be an opportunity to do a feasibility test, generate some data, work with our team, put any risks to rest. We communicate openly, all issues are addressed, unparalleled attention to detail. In fact, we're obsessed. And after one experience, we'll quell any hesitation. And when you work with Peapod Bio, you can actually take a vacation. Whether benefiting from our label-free services or beyond, or even requesting a quote, you'll be surprised how fast we respond. We can develop new assays, not in weeks, but days. Our team's catalytic, like Peapod oxidase. Many times we send you data in less than 24 hours, but we don't cut corners in our core, our superpowers. And while technically a startup, our minds are older. And when life science budgets get colder, we shoulder every target, every challenge. We have learned to manage. No fake claims to brandish. We give you every possible advantage. We use technologies that drive discovery projects, and our polymeric enrichment arrays is just one of our concepts. Our expertise is in biochemistry and assay development to ensure your project is successful and biologically relevant. We can develop any assay. We live and breathe the fundamentals, and our team has the highest standards and higher still potentials to identify leads with any form of inhibition Peapod Bio's capabilities will put you in first position. And if you need binders like Protax or glues, we develop ASMS assays as if we were in your shoes. We complement that with optical assays and world-class libraries, with no surprises in data, time, or cost, unlike others where the price varies. So if you'd like to screen with our label-free and beyond solutions, you can screen our library and/or your institutions. And what about other sources? I could go on and on. We also accept compounds from Psi, Wuxi, and Pharmaron. Now, sometimes with mass spec, analytes may be hidden and make you leave out key buffer components that are strictly forbidden. In detecting small molecules, there could be interference with matrix that may steer you towards another assay, not wanting to take risks. And then there are those substrates and products with an unresolvable mass. Well, Peapod Bio developed solutions, so let's get down to brass tacks. In case you're new to the surface chemistry game, let me explain our surfaces that became our claim to fame. Alkane phthalates on gold, first described by Nuzzo and Alara, then pioneered by George Whitesides, a professor at Harvard. The use of polyethylene glycol prevents unwanted protein absorption, and I developed similar chemistries as a grad student, let's not forget that portion. But fast forward 20 years and we're still innovating at the surface, and it's still incredibly valuable, and that's why we offer it as a service. Polymeric enrichment arrays, or what we call Ps, we can use them to assay any target in any area of disease. You may be asking how we deal with ion suppression. It's the bane of existence in the mass spec profession, with no LC or OPP to remove buffering components, and desalting steps take too much time and steal those precious moments. You can also benefit from minimal target consumption and screen 100,000 compounds per day. That's an accurate assumption. So if you want to optimize your assay for whatever your target needs, our RapidFlex MALDI PharmaPulse is best served with Ps. And if your target's not an enzyme, we can also measure binding with the fastest ASMS workflow that straightens a road that can be winding. And today I'll highlight creative solutions using our arrays to resolve overlapping peaks, and it might just take your breath away. So how do we do it? How do we utilize this technology? Well, listen up closely, and I'm sure that you can follow me. It starts like any assay in a high-density well plate. Add enzyme to compounds, initiate with substrate. After incubation, we quench the reaction, transfer them to the arrays, no need for an LC fraction. They purify your analytes of interest within seconds, so we can use whatever buffer is best, whatever your target reckons. Remember that the surfaces are specific and tailored and prevent unwanted protein absorption with that stable monolayer. And it's well known, this chemistry. It's been described extensively. We use it inexpensively and we understand it comprehensively. And after we immobilize your substrates and products to the array, we rinse the plate, we add a matrix, and it's stable the whole day. The MALDI laser desorbs only the analyte we want to generate peaks, and we can screen 100,000 compounds in a day, not weeks. Next, we read the plate on our RapidFlex from Bruca. My accent's wicked strong here, like a Boston Mass State trooper. Let's run through a case study with the enzyme sortase. It's involved in bacterial infections, but it won't bring sores to your face. This is an enzyme that cleaves a peptide and then ligates to disrupt the cell's immune response like a bacterial spygate. It's implicated in virulence and antibiotic-resistant MRSA, so we decided to run a screen and find inhibitors to reverse it. The traditional way to assay this is by using a fret pair, but it's plagued by caveats more prevalent than my shedding dog's pet hair. One challenge is the opportunity for optical interference. Now, you could spend months triaging hits and chalk it up to perseverance, but we designed an assay using Ps and MALDI, and we orchestrated it beautifully, like strings and Vivaldi. So if you'd like to learn more about assay development and the screen, stop by our poster and you'll see what I mean. But for now, I'd like to highlight some unique benefits to rule out false positives that are a screen's biggest nemesis. Our label-free readout identified several promising hits. Had we used the optical assay, these would have been missed. And what happens if a compound has a primary amine? That's another false positive to come out of that FRET screen. But mass spec in P's can resolve multiple products. It's like fine art, but not abstract, like Jackson Pollock's. And had we only focused on a single product peak, we would have also called many compounds as hits, so to speak. But using our Rapiflex MALDI's analytical prowess, we had it calculate potential compound reactions, and that allowed us to rule out false positives through the primary amine, and it saved us a bunch of money and time with this screen. So I encourage you to check out our poster. It's tomorrow from 2 to 3 p.m. And I promise someone else will be there and he won't speak like Eminem. While biochemical assays are routine and built into our genetic code, there's another application of P's discussed in this next episode. There are many ASMS techniques that are used quite commonly, but they require slower steps like size exclusion chromatography. To increase the speed, libraries are often compressed. Up to thousands of compounds per pool is what I've seen in some tests. And other approaches that utilize similar surfaces force desorption of a monolayer that was designed for other purposes. Then compounds that overlap with that standard will skew the data. You won't see the peaks now, but you'll pay for it later. So we designed a workflow that's up to 100 times faster. Incubate target and compounds in solution first and immobilize them after. We wash away non-binders and buffers in less than 3 seconds. Again, we can use whatever buffer is best, whatever your assay beckons. Next, we add a matrix which can serve as an internal standard. You may have questions about this, and I'll make sure they're answered. The MALDI data informs on compounds that are potent and specific, and the reference surface serves as a counter target. Another reason it's terrific. And in case you need binders that may be weaker, we can screen much higher compound concentrations, including tens of micromoles per liter. Now, since we don't desorb the surface, we can optimize conditions for the analytes of interest that we want on exhibition. Those familiar with MALDI know it's a soft ionization technique, but we develop solutions that fragments matrix into reproducible peaks. It generates a fingerprint we can use for quantitation. That gives us multiple standards to choose from, and this slide shows an illustration. And because these peaks are consistent and reproducible, if a compound peak overlaps, our approach makes it deducible by calculating the ratios of the different fragment peaks. We can identify any compound, including those playing hide and seek. And that's how our technology can find anything that binds, which makes our ASMS approach proprietary and one of a kind. But wait, there's more. There's another unique solution. It's just another way we're innovating the ASMS evolution. We can also measure peptides and other binding ligands by tailoring the standards and settings. But wait, the plot thickens. You see, a challenge in proximity testing is detecting the ternary complex between a target, the small molecule, and its binding accomplice. You can either measure compound binding or stick to proximity. But our approach can do both with technical efficiency. We include multiple standards, one for the peptide, one for the compound, and we analyze them in parallel because we're not one to mess around. And that's how our arrays can bring into vision a variety of analytes that traditionally stay hidden. With the last few minutes, I'd like to leave you with a teaser to make you become a true Peapod Bio believer. There are thousands of enzymes whose substrates and products differ by 1 or 0 Daltons. Now, typically, that would block us from using mass spec to analyze these reactions. But our arrays can achieve it with a simple transaction. Normally, I don't recommend a coupled enzyme system, although sometimes for optical assays, I know we can't resist them. But no matter the readout, when multiple enzymes are present, a compound in that reaction could inhibit the first or the second. Then you have to develop even more orthogonal assays after you just got done telling me how your budget's gone sideways. So after we immobilize the substrates and products to the array, we wash away the reaction and the compounds. It's a step we do anyway. And if we try to read the surface now using mass spec, we'd likely see only a single peak with one hidden below deck. Instead, we have an opportunity to run a second reaction in an optimized buffer absent the compounds, and that's the main attraction. And because our biologists are always creative in their design, so the substrates and products react differently, then we can define the enzyme reaction progress cleanly with confidence because each peak stands out instead of being anonymous. Take, for an example, the reaction of adenosine to inosine. With a 1 Dalton mass shift, there's no way mass spec data can be that clean. But we designed a reaction where we can simply assign each peak that stands out and that R squared 0.9999. Thank you for your attention and putting up with this presentation style. It's just another example of how Peapod Bio goes the extra mile. We take every assay seriously. Every detail is considered. And our goal is to ensure that novel drug leads are rapidly delivered. And in case some of you think I wrote this with help from ChatGPT, I promise you, I didn't. That's not our AI strategy. So remember, the next time you need an assay and mass spec is what it needs, our RapidFlex MALDI PharmaPulse is best served with peas. Thank you for your attention. Come and check us out at booth 1129. And in case you can't make it, you can reach out anytime.